Discuss about the topic gram-positive bacteria that comes under the bacteriology. So, in this video, we mainly discuss about the gram-positive bacteria because unit 1 that was in bacteriology is about the gram-positive bacteria. What are the characteristics of the gram-positive bacteria, how we characterize the gram-positive bacteria, how we differentiate between the gram-negative and gram-positive bacteria. So this all we discuss in this video. Okay. So first of all, what is the bacteriology? What is the bacteriology and why we study this bacteriology, right? So bacteriology is nothing but it is the branch of biology in which we study about mainly the bacteria, right? Mainly the bacteria. From name it's clear that bacteriology, bacteria, and logy means we study about the bacteria. So in this bacteriology, what happened? We characterize the morphology, ecology, genetics, biochemistry of bacteria, as well as many other aspects that are related to bacteria, right? What are the morphology of the bacteria? What is the ecology of the bacteria? What are the genetics of the bacteria, means what type of genetic material contain that bacteria, means like DNA, RNA, and what are the biochemistry of the bacteria, what are the chemical by which this the cell wall are made up of, what are the chemical by which cell membrane are made up of, what are the differentiate between gram positive and gram negative bacteria. So this we all discuss about in this bacteriology, right? So this subdivision of microbiology involves the identification, classification, and characterization of the bacterial species, right? This subdivision of microbiology involves the identification, classification, and characterization of bacterial species. So first of all, what are the history? So the discovery of the connection of the microorganism. So the discovery of the connection of microorganism to disease can be dated back to the 19th century when German physician that Robert Koch introduced the science of microbiology to the medical field, right? Science of microbiology to the medical field. Robert Koch was the first scientist who introduced the science of microbiology, means microbes. They introduced the microbes like bacteria, virus, protozoa. This all are of microbes that Robert Koch introduced, right? So he identified the bacteria. Robert Koch identified the bacteria that cause the infectious disease and process of fermentation in disease. Right? So another French scientist like Louis Pasteur developed techniques to develop or produce a vaccine. Another scientist what her name? Louis Pasteur. Louis Pasteur was another scientist who developed the technique to produce the vaccine for for cowpox, chickenpox. They introduced the vaccine. So both Koch and Pasteur play a role in improving antisepsis in medical treatment. Both Koch and Pasteur play a role in improving antisepsis in medical field. Clear? So what happened? Robert Koch was the first scientist who introduced the microorganisms, right? Robert Koch was the first scientist who introduced the microorganism in the field of medical science, and they identified the bacteria as a causing agent of infectious disease, right? And another scientist, Louis Pasteur, who developed techniques to produce vaccine. Now, how we identify the gram-positive bacteria and how we differentiate this is the gram-positive bacteria and this is the gram-negative bacteria. So, there are techniques. There are a staining method by which we differentiate the gram-positive bacteria and gram-negative bacteria. So, in gram staining method, in bacteriology, gram-positive bacteria are bacteria that give a positive result, that give a positive result in gram stain test, which is traditionally used to quickly classify bacteria into two broad categories according to their type of cell wall, which stain they contain, right? Like gram positive bacteria contain the gram stain and they appear as a purple color, whereas when wash the gram negative bacteria, the gram stain was be removed, right? So that's why they appear as a pink in color. So by these techniques, we differentiate the gram positive bacteria and gram negative bacteria. So in bacteriology, gram positive bacteria are bacteria that give a positive result in gram staining result, which is traditionally used to quickly classify bacteria into two broad categories according to their type of cell wall. So gram positive bacteria take up the crystal violet stain used in the test and then appear to be purple color when seen through a optical microscope. Gram positive bacteria take up the crystal violet stain used in the test and then appear to be purple colored when seen through an optical microscope. This is because the thick peptidoglycan layer in the bacterial cell wall retain the stain after it is washed away from the rest of the sample in the decolorization stage of the test. So gram positive bacteria retain the crystal violet stain that used in the gram staining techniques and then appear to be purple color when we see that gram positive bacteria under the optical microscope. Because the gram positive bacteria having a thick peptidoglycan layer, whereas gram negative bacteria having a thin peptidoglycan layer. That's why gram positive bacteria retain that gram staining and gram negative bacteria remove that stain. Clear? So in other hand, gram negative bacteria cannot retain the violet stain after the decolorization step. Alcohol used in this stage degrades the outer membrane of gram negative cells. What they degrade the outer membrane of gram negative stain and they making the cell wall more porous and incapable of retaining the crystal violet stain and incapable of retaining the crystal violet stain because they having a thin peptidoglycan layer. That's why their peptidoglycan layer is much thinner and sandwiched between inner membrane and bacterial outer membrane. What happened in gram negative? This is the peptidoglycan layer and this peptidoglycan layer are sandwiched between the inner layer membrane and outer membrane of bacteria. So this peptidoglycan layer is in gram negative bacteria was thin and the bacterial outer causing them to take up the counter stain and appear red or pink. See, this is the gram negative bacteria and gram positive bacteria. This is the gram positive bacteria, whereas this is the gram negative bacteria appear pink in color and this appear violet in color sphere, right? This is violet in color are gram positive bacteria and pink in color was a gram negative bacteria. This is the cell wall structure of gram positive and gram negative bacteria. See, peptidoglycan layer. This red color was a peptidoglycan layer, right? In gram positive bacteria, this peptidoglycan layer was a thick, whereas in gram negative bacteria, this peptidoglycan layer was thin, right? So this is the plasma membrane, this is the periplasmic space, and this is the peptidoglycan layer. Whereas in gram negative bacteria, this is the plasma membrane, right? This plasma membrane, then periplasmic space are present, then peptidoglycan layer are present, then again periplasmic space are present, then outer membrane are present. Means this peptidoglycan are sandwiched between the inner membrane and outer membrane of bacteria, right? This is the main difference between the gram positive and gram negative bacteria. So gram positive bacteria, classification of gram positive bacteria. Gram positive bacteria are classified into cocci and bacilli, right? Cocci and bacilli. Bacilli, Corynebacterium, Clostridium, Listeria, and Bacillus. Corynebacterium, Clostridium, Listeria, Bacillus, right? And cocci, Staphylococcus, and Staphylococcus cattle is negative, cattle is positive. According to the reaction or taste, this cocci are classified into two types, that is Staphylococcus positive and Staphylococcus negative, right? So Staphylococcus positive, which show positive reaction in catalase, are two types. S. aureus, Staphylococcus aureus, coagulase positive and coagulase negative. So S. epidermis, Staphylococcus epidermis, novobiocin sensitive, and Staphylococcus saprophyticus, novobiocin resistant. Clear? Novobiocin resistant, right? And Staphylococcus cattle is negative, beta hemolytic, gamma hemolytic, and alpha hemolytic. Beta hemolytic are clear and alpha hemolytic are also, sorry, beta hemolytic are clear and alpha hemolytic are green in shape, green in color, right? In this, what example? Pyrogenous, group A, bacitericin sensitive, right? Group B, bacitericin resistance, right? So this is the antibiotic by which bacteria are resistant and bacteria are sensitive to them. According to that, we classify this bacteria, right? So gram positive bacteria are two types, coccus, in two shape, that they are coccus or bacilli. Coccus are classified into two types according to their catalase positive reaction or negative reaction, right? So catalase positive are a coagulase positive and coagulase negative. Staphylococcus catalase negative are classified into three types: alpha hemolytic, beta hemolytic, and gamma hemolytic, right? And in bacillus, what are the example? Corynebacterium, Clostridium, Listeria, and Bacillus. Clear? So this is the cell wall structure. This is the cell wall structure of gram positive bacteria. See, this is the N-acetyl glucosamine. This the cell wall are made up of NAG and NAM. NAG and NAM. NAG means N-acetyl glucosamine and NAM means N-acetyl muramic acids. And this acetyl NAG and NAM are joined with each other with two tetrapeptide chain that cross linked by peptide. Two tetrapeptide chain that are cross linked by peptide, right? And this cross link by cross linking, they form a peptidoglycan layer. So plasma membrane showing lipid bilayer. Plasma membrane are lipid bilayer. This is the plasma membrane and this plasma membrane are lipid bilayer. Clear? So this is the peptidoglycan layer. This is the plasma membrane showing lipid bilayer, and this peptidoglycan layer are made up of NAG and NAM, and this NAG and NAM are joined with the two tetrapeptide chain that cross linked by peptide. Now pathogenesis. What happened in pathogenesis in gram positive bacteria? So in this classical sense, 6 gram positive genera are typically pathogenic in human. What? Typically pathogenic in humans. 2 of these are Streptococcus and Staphylococcus, that are cocci. And the remaining organisms are bacilli, that are rod shaped and can be subdivided based on their ability to form spores. Based on their ability to form the spores. The non-spores formers are Corynebacterium and Listeria, that are Coccobacillus, whereas Bacillus and Clostridium produce spores. The spore forming bacteria can again be divided based on their respiration. That is, Bacillus is a facultative anaerobe while Clostridium is an obligate anaerobe. Bacillus is the facultative anaerobe, whereas Clostridium is obligate anaerobe. Means they survive only when oxygen are not present, whereas Bacillus survive in both, oxygen are present or not present. But they are anaerobe, means they require no oxygen, but if oxygen are present, then also they survive. That's why they are facultative anaerobe. Also Rathibacter Lefsonia, and Clavibacter are 3 gram-positive genera that cause plant disease. The gram-positive bacteria are capable of causing serious and sometimes fatal infection in newborn infants. So novel species of clinically relevant gram-positive bacteria also include Catibacter oncogenesis, which is an emerging pathogen belonging to Firmicutes. Right, so mainly 6 gram genera are typically pathogenic, and that in 6 gram positive genera, 2 are a cocci, that is Staphylococci and Streptococci, right? Staphylococci and Streptococci, and remaining 4 genera are bacilli that are rod shaped and can be divided on the formation of their spores, right? Because with the help of a spore, they cause a disease. The non-spore former are Corynebacteria and Listeria. Means this Corynebacteria and Listeria are not too much harmful, not too much pathogenic. But Bacillus and Clostridium that produce spores, they are too much pathogenic to human, right? So how this bacterial transformation will be takes place? How they divide, or how their transformation, means how the genetic material will transfer from one bacteria to another bacteria. So transformation is one of the 3 processes of horizontal gene transfer in which exogenous genetic material that are passes from a donor bacterium to recipient bacteria. The other 2 processes that are a conjugation, transfer of genetic material between 2 bacterial cells in direct contact, and transduction, injection of donor bacterial DNA by a bacteriophage virus into a recipient host bacteria. In transformation, the genetic material passes through the intervening medium and uptake is completely dependent on the recipient bacteria. Clear? So transformation is one of the 3 processes by which the genetic material or plasmid will be transferred from one bacteria to another bacteria. Another is the conjugation, by which also the genetic material will be transferred from one bacteria to another bacteria. When genetic material will transfer from one bacteria to another bacteria, means one bacteria contain the plasmid and this bacteria not contain the plasmid, this bacteria is called as a F minus plasmid, and this is called as a F plus plasmid. So what happened? This bacteria transfer their plasmid to this bacteria, then this bacteria also called as a F plus, and this is also a F plus. Right. So injection of donor bacterial DNA by a bacteriophage into a recipient host cell, right? So in transformation, the genetic material passes through the intervening medium and uptake is completely dependent on the recipient bacteria. In transduction, what happened? In transduction, bacteriophage or bacteriophage that are a virus, which infect the bacteria. So bacteriophages are used in transduction process for the transfer of genetic material from one bacteria to another bacteria. Clear? So Staphylococcus. Staphylococcus is a gram-negative bacteria, already you know, and from name it is clear that Staphylococcus are a cocci shape and means rod sphere shaped, right? So it is frequently involved in nosocomial and opportunistic infection. And different species of Staphylococcus are Staphylococcus aureus that lives in respiratory tract and skin, Staphylococcus epidermis that lives on skin and mucous membrane, Staphylococcus hominis that live around sweat glands, Staphylococcus capitis that live on scalp, face, external ear, Staphylococcus saprophyticus that lives on skin, intestine and vagina. So this Staphylococcus is a gram-positive bacteria, and this Staphylococcus are pathogenic, highly pathogenic for human beings, right? So it frequently involved in nosocomial and opportunistic infection. Staphylococcus lives in respiratory tract. Remember this, right? Epidermis lives on skin and mucous membranes. Remember this. Staphylococcus hominis lives around the sweat gland. Staphylococcus capitis lives on scalp, face, external ear, right? Scalp, face, and external ear. Staphylococcus saprophyticus lives on skin and intestine and vagina. So now pathogenesis. Present in most environments frequented by humans. Carriers rate for healthy adult is 20 to 60%. Carriers is mostly in anterior nares, skins, nasopharynx, and intestine. And predisposition to infection include poor hygiene, nutrition, tissue injury, pre-existing primary infection, diabetes, and immunodeficiency, right? So this pathogenesis present in most frequented by humans, and they carry a human adult by 20 to 60%, and carriers is mostly in anterior nares, skins, and nasopharynx and intestine. Predisposition to infection includes poor hygiene and nutrition, tissue injury, pre-existing primary infection, diabetes, immunodeficiency, right? So now, what are the classification based on the pigment production and based on the pathogenesis and based on coagulase production? So based on the pigment production, the gram-positive bacteria are classified into Staphylococcus aureus that appear as golden yellow pigmented colonies, Staphylococcus albus, white colonies, lemon yellow colonies, right? All and citrus from name it's clear, Staphylococcus citrus from name clear, that's lemon yellow colonies. And pathogenic includes only one, that is Staphylococcus aureus, and non-pathogenic includes Staphylococcus epidermidis, Staphylococcus saprophyticus, Staphylococcus albus, and Staphylococcus citrus that appear as lemon yellow color, right? So Staphylococcus citrus are non-pathogenic. Mainly pathogenic are Staphylococcus aureus are most pathogenic, and Staphylococcus epidermis are Staphylococcus saprophyticus, Staphylococcus albus, Staphylococcus citrus, Staphylococcus hominis are non-pathogenic. And based on the coagulase production, that is coagulase positive and coagulase negative. In coagulase positive, Staphylococcus aureus are present, and in coagulase negative, Staphylococcus epidermis, Staphylococcus saprophyticus are present. Okay? So these all are about the gram-positive bacteria. How we classify the gram-positive bacteria? First of all, what is the bacteriology? In bacteriology, we mainly discuss about the bacteria and what are the characteristics of the bacteria, how we classify the bacteria. This all, what are the morphology, how they transfer from one transformation process, their reproduction process. So this all we discuss in bacteriology, or right? In gram-positive bacteria are classified. Gram-positive bacteria are classified according to the gram staining method, right? The bacteria which retain the stain, gram stain, that called as a gram-positive bacteria, and the bacteria which not retain the gram-positive stain, that called as gram-negative bacteria, right? So gram-positive bacteria are having a thick peptidoglycan layer, that's why they retain the gram-positive stain, whereas gram-negative bacteria having a thin peptidoglycan, that's why they not retain the gram stain, right? And that's why they appear as a pink in color, and gram-positive bacteria appear as a violet in color. The gram-positive bacteria are classified into two types according to their shape, cocci and bacillus. In cocci, staphylococcus and streptococcus, right? They are positive or negative. They are according to the catalyst, positive or negative reaction. In bacillus, they are rod shaped. Pathogenesis, mainly 6 genera of the gram-positive bacteria are mainly pathogenic, right? In bacillus, spore forming bacteria are pathogenic, and non-spore forming bacteria not too much pathogenic, right? So this all are what are the process of transformation. There are three processes: that transformation, transduction, and conjugation, by which they transfer their genetic material from one bacteria to another bacteria, right? So this all are about the gram positive bacteria. So today we stopped here. Hope this video are clear to you. Thank you.
So students, in this lecture, we mainly discussed about the gram-negative bacteria that was under the bacteriology topic. Okay. So first of all, what do you mean by bacteriology, right? So bacteriology is nothing but it is from name it's clear that bacteriology is a branch of science in which we deal or discuss about the bacteria, right? Actually, bacteria are of two types according to the gram staining techniques. According to the gram stain. So bacteria are of two types, gram positive bacteria and gram negative bacteria. So in this bacteriology, we discuss mainly about the gram positive bacteria. One unit is the unit first is about the gram positive bacteria. Unit second is about the gram negative bacteria. So in this lecture, we discuss mainly about the gram negative bacteria that was under the unit 2. So first of all, what is the gram negative bacteria? So gram-negative bacteria are bacteria that do not retain the crystal violet stain used in the staining method techniques, right? So what gram staining is the process of staining by which we can differentiate the gram-positive bacteria and gram-negative bacteria, right? So gram-positive bacteria having a thick peptidoglycan layer in their cell wall, right? Thick peptidoglycan layer. Whereas gram-negative bacteria having a thin peptidoglycan layer in their cell wall. Clear? So that's why gram-positive bacteria can retain the stain, gram stain. That's why they appear as violet color. Whereas gram-negative bacteria doesn't retain that gram stain, right? So that's why they appear as pink in color because they stain with the safranin, right? So this is the difference between the gram positive and gram negative bacteria. So gram negative bacteria are bacteria that do not retain the crystal violet stain used in the gram staining method or bacterial differentiation. So they are characterized by their cell envelope, which are composed of a thin peptidoglycan cell wall that sandwich between the inner cytoplasmic cell membrane and outer bacterial outer membrane, right? So peptidoglycan layer are sandwiched between the cytoplasmic membrane, means plasma membrane, or outer bacterial outer membrane. That was the main cell wall, right? So gram negative bacteria are found virtually all environment on earth that support the life. Okay? So gram negative bacteria include the model organism that is E. coli, that is Escherichia coli, as well as many pathogenic bacteria such as Pseudomonas, Chlamydia, Yersinia. They are an important medical challenge as their outer membrane protects them from many antibiotic detergent that would normally damage the inner cell membrane. But this gram negative bacteria like E. coli, Chlamydia, Yersinia. So this type of pathogen having a cell wall structure which protect which protect that microbes from the antibiotic that we eat when we have an infection, and we eat that antibiotic, then that antibiotic doesn't damage this microbes, because this microbes are having a cell wall structure, and that cell wall protect them from protect this protect this microbes from the antibiotic. Clear? And detergent that would normally damage the inner cell membrane, and an antimicrobial enzyme produced by animals that form part of the innate immune system. So additionally, the outer leaflet of this membrane comprises a complex lipopolysaccharides whose lipid A component can cause a toxic reaction when bacteria are lysed by immune cells. When bacteria are lysed by the immune cells. So this toxic reaction may lead to low blood pressure, respiratory failure, reduced oxygen delivery, and lactic acid acidosis, and manifestation of septic shock. So this is the structure. This is the structure of the bacteria, right? So bacteria having a plasmid, chromosome, ribosome, cell wall, cell membrane, and flagella, right? So flagella help in the movement. Flagella help in the movement of the bacteria, right? Cell wall protect the bacteria from the adverse condition, like when we eat antibiotic or when temperature will increase. So this cell wall protect this bacteria from the adverse condition. Chromosome contain the genetic material, that DNA. Plasmid is the extra chromosomal material that are present in bacteria and which help in the transformation process, right? So bacteria contain a singular circular piece of DNA, teeny circular pieces of DNA called, called plasmid, ribosome, right? How the bacteria will reproduce? How this gram-negative bacteria will be reproduced? So bacteria will be reproduced by asexual method, that is the process mitosis using binary fission, using the binary fission. For example, E. coli, the doubling time of E. coli is 20 minutes. After each 20 minutes, the number of the E. coli will be double from the initial number, right? So this is the bacteria, right? This is the prokaryotic chromosome, plasma membrane, cell wall. This bacteria will duplicate, having duplication of the chromosome. Then chromosome will be duplicated. When chromosome will duplicated, continue growth of the cells, and when cell will be continue with the growth, the division, they divided into two cells, and this process is known as the binary fission. Yeah. Bacteria reproduced by sexually using the conjugation process, right? With the help of conjugation process. So bacteria exchange plasmid DNA. This is how bacteria become antibiotic resistant. When by this process, bacteria will become the antibiotic resistant. See here, this is the bacteria. This is the donor F plus cells, right? This is the recipient F minus cells. This F plus cells having a plasmid, F plasmid. This F plus cells having F plasmid, whereas this recipient cells, F minus cells, doesn't having the plasmid. Yeah? So what happened here? This plasmid will go inside this F minus cells. So when this plasmid go inside this F minus cells with the process of the conjugation, this F minus cell also converted into a F plus. Yeah. So the F plus donor cell containing an F plasmid is capable of synthesizing a sex pilus. Right? This is the pilus, right? The sex pilus contact the recipient F minus cells. This sex pilus comes in contact with the F minus cells. The plasmid is activated for transfer when an endonuclease cleaves one strand of DNA the origin of transverse endonucleases came and cleave this plasmid at one strand, so that this plasmid go inside the F minus cells. So the sex pilus retract and pulls the donor and recipient cells together. The F plasmid is transferred as a single-stranded DNA molecule. This F plasmid go and transfer into a single-stranded inside the single-stranded DNA molecule, right? So this is the F plasmid transfer. The complementary strand to both F plasmid strands are synthesized in donor and recipient cells, and both cells are become F plus and synthesizes the sex, right? So this both cells become a F plus cells, and both cells have a capability to synthesize the synthesize the sex pilus, right? So that this sex pilus help in the conjugation process, right? So bacteria have cell wall made up of peptidoglycan. The bacteria have the cell wall which is made up of peptidoglycan, and this peptidoglycan have a polysaccharide linked with chain of amino acids. Polysaccharides linked with chain of amino acids. So this may be covered with an outer membrane of lipopolysaccharides, chain of sugar with a lipid attached. So some bacteria have a gelatinous layer called a capsule. What? Called a capsule surrounding the cell wall and form thick wall endospore and around the chromosome. When they are exposed to harsh conditions like drought, high temperature, this type called botulism, and they produce an endospore to protect the microbes, right? To protect the cells. So bacteria having flagella, and flagella help in the locomotion. Pili, they also having a pili. See, this is the pili. This is small is the pili, right? Flagella, this long structure, thread-like structure is the flagella, and this small structure is the pili. So pili are short, thicker outgrowth that helps us to attach to the surface. They help in attached to the surface, and this flagella help in the locomotion. So bacteria can be moved by air, water, currents, and on any surface such as clothing, hands, or any object. Yeah. So there are three bacterial shapes, that is rod shape, that called bacilli, sphere shaped, that are called coccus, spiral shaped are called spirilla, right? This is the spherical shape that called cocci. This is the spiral shape that is spirilla, and this is rod shape that is a bacillus. So there are three type of shape of the bacteria, that are rod shaped, sphere shaped, and spiral shaped. So gram staining, how this bacteria, what are the gram staining and how this bacteria will differentiate it? Important in medicine because provide information for treatment of bacterial disease. First, we have to identify which type of bacteria this is, right? So bacteria stain either gram positive. If they stain the gram stain, means they appear as violet in color, they are known as gram staining, and they appear as a purple or violet color. Or gram negative bacteria having a pink color. So gram positive tends to respond to penicillin and like antibiotic. Gram negative responds to type of antibiotic unrelated to penicillin. So gram negative cell wall having multi-layer and more complex than gram positive cell wall. Clear? Multi-layered and more complex than gram positive cell wall. Peptidoglycan of gram negative bacteria is thin and comprises only 10% or less of cell wall. Outer membrane lies outside the thin peptidoglycan layer. Most abundant protein is brown lipoprotein. So multi-layer and more complex than a gram-positive cell wall. Gram-negative cell wall having a more complex. Because there are more layer of cell wall are present in gram-negative bacteria. So peptidoglycan of gram-negative bacteria is thin and comprises 10%, only the 10% or less of cell wall. Outer membrane lies outside the thin peptidoglycan layer. Gram staining process, crystal violet dye, purple, iodine decolorizes them. After that, alcohol, pink, pink, and counter stain, that is saffron. Okay, this is the process. First, they stain with the gram violet, crystal violet dye. If they are gram positive bacteria, they stain this. Iodine decolorize this purple color. If they are gram negative bacteria, this purple color will be decolorized. And if they are gram positive bacteria, then this purple color will be not decolorized. Right? After they washed with alcohol, then counter is stained with saffron, and they appear as pink gram negative bacteria. So gram staining process are, are flood air dried heat fixed smear of cells for 1 minute with crystal violet staining reagent, right? Crystal violet staining reagent for 1 minute. Then wash slide in a gentle and indirect stream of tap water for 2 seconds. Flood slides with the moderate gram iodine. Wait 1 minute. Then wash the gentle and indirect stream of tap water for 2 seconds. Then the decolorizing agents, that is acetone alcohol decolorizer, wait 10 to 15 seconds or add drop by drop to slides until decolorizing agent running from the slide runs clear. After that, counter saffroning and wait for 30 seconds to 1 minute and wash the slide gently and indirect stream of tap water until no color appear in the effluent and then blow dry with absorbent paper. After that, observe the result of the staining procedure under oil immersion using a bright field microscope. So gram-negative bacteria will stain pink, red, and gram-positive bacteria will stain purple or violet in color. Clear? This is the procedure, flow-through procedure. Bottom of the biofilm slide clean top edges slides about 2 mm. Build a bodies of petroleum jelly on the top and bottom of a cover slip. Cover slip with petroleum jelly. Biofilm on slide with cover slip. Here, add crystal violet, wait 30 minutes. Wash with water, add gram iodine, wait 1.5 minutes, decolorize with alcohol, then wash with water, stain with saffron dye, wait 30 seconds, wash with water, examine under oil immersion through the cover slips. Right? So gram-positive bacteria purple because large amount of peptidoglycan in cell wall. Polymer consists of sugar and amino acids that form a mesh-like layer outside the plasma membrane of bacteria. So gram-negative bacteria, pink because lipopolysaccharide membrane is removed by alcohol. What? Lipopolysaccharides membrane are removed, right? By alcohol and thin peptidoglycan layer allow dye to escape. Clear? So LPS is a large molecule. LPS is the large molecule consisting of a lipid and the polysaccharides joined by a covalent bond. So this is the OA specific side chain, and this is having a lipopolysaccharide. This is a periplasmic space and peptidoglycan, and this is the thin as compared to gram-positive bacteria. That's why they remove the crystal staining, right? They remove the crystal staining. So gram-negative bacteria constitute the largest group of human pathogen, right? Due in part to the presence of lipids A in the bacterial cell wall, trigger fever, vasodilation, inflammation, shock, and dissemination, intravascular coagulation, right? So blood clot within the blood vessel. Almost every gram-negative bacteria that can breach the skin or mucous membrane grow at 37 degrees Celsius and evade the immune system and can cause disease and death in human. Enterobacteriaceae. Member of the intestinal microbiota of most animals and humans, right? Members of the intestinal microbiota of most animals and humans, ubiquitous in water, soils, and decaying vegetation. Enteric bacteria are the most common gram-negative pathogen of humans. Enteric bacteria are the most common gram-negative pathogens of the human. Clear? So general properties of Bacteriaceae is the gram-negative bacilli. They having a rod-shaped structure. Aerobes and facultative anaerobes grow on ordinary media, ferment glucose with production of acid or acids and gas. They reduce the nitrates to nitrites. Catalase positive, these are called catalase positive, oxidase negative, motile or non-motile. They having maybe flagella or they not maybe not having a flagella. So Bacteriaceae is the rod-shaped structure. They are aerobes and facultative anaerobes, grow on ordinary media, ferment glucose, reduce nitrates into nitrites. Yeah? They are catalase positive, oxidase negative, motile or non-motile. So work by preventing cell wall formation and breaking up cell membrane on disruptive chemical process. This is the function of the antibiotic. But gram-negative bacteria having a cell wall, so cell wall protect the microbes against the antibiotics. So antibiotic cannot treat viral infection. There are three types of bacteria based on how they obtain energy are heterotrophs, photosynthetic, and chemoautotrophs. This is the anthrax bacteria, right? So this is the diagram or figure photos of the diseases that are caused by the gram-negative bacteria or some other bacteria, because Streptococcus is the gram-positive bacteria, right? So how this necrotizing fasciitis, light skin, especially important in Streptococcus problem, lower leg, dark skin, right? This is the how the bacteria cause the disease, right? So today we stopped here. Hope this video are clear to you, right? So today we stop here. Thank you.
In this video, we discuss about the different types of biochemical test. First of all, you all already know what is the biochemical test. In everyday life, generally, we know or discuss about the different types of biochemical tests like lipid profile test, renal test, glucose tolerance test, sugar diabetes test. So there are different type of tests are presented or they are in daily life. So every person go through the biochemical test. There are different sample taken by the technician for the biochemical test, like they collect sample like blood for the GTT, right? For the GTT test, they collect sample of blood. For renal test, for renal function test, they collect sample blood or urine. So the sample depend upon the what type of tests are performed. What type of tests are performed by the person. So according to the test, sample are collected. So first we discuss about the introduction. What is the clinical? Actually, the biochemical tests are generally used in a clinical biochemistry. This is the one unit in the introduction to biochemistry of BVMLT-204. So first of all, introduction. Why these tests are used? Why we use the biochemical test? Why we perform the biochemical test? For the to know the functioning of the body, to know the functioning of the body. So for know the functioning of the body, our body perform proper function or not, is the metabolic process will take place regular, proper or not. For know this proper functioning of the body, we use this biochemical test. If there are some problem in our body, then doctor prescribe like go and test the lipid profile test, cholesterol test, GTT test, like this. Before the operation, before the any operation, like any operation, doctor prescribe first go and test your body test, like lipid profile test, sugar test. If your sugar is increased or high or low, the operation will not take place. If lipid function test, the the report of the lipid function test is not proper, is not proper, then also, if not proper, then also operation will not take place. So for the operation, for the any type of operation, the proper functioning or proper metabolic reaction of our body will be normal. Then after operation will be takes place. For eye operation, for um, for the stone operation, any type of operation. If your sugar level will increase, then doctor told you go and control your sugar. After that, you come for the operation. So biochemical tests are very important. Biochemical investigation are involved in every branch of clinical medicine, right? In every branch of clinical medicine, biochemical investigation will be takes place. It's either microbiology, it's either microbiology or biochemistry, right? Or immunology, any branch of the clinical medicine, the biochemical test will be takes place. The result of test may be used in diagnosis in the monitoring of the treatment. If your sugar is high, if your sugar is high, then doctor prescribe you for the diabetes, right? And they monitor the treatment according to the level of the glucose in your body. According to that, doctor monitor the treatment. Screening for disease or in assessing the prognosis. Which type of disease is in your body? That disease is identified by the biochemical test. What? That type of disease is identified by the biochemical test. Clinical trial of new drugs. There are many of the research will be going on the invention of the new types of drugs, right? Many invention or research is going on the invention of new types of drug. So how this new type of drug will be identified? This drug, for trial, this drug injected into a person, right? After that, biochemical test will be performed by this person, so that we identify the properties or functioning properties or functioning, right? Properties or functioning of the new drug. Clear? So, in the clinical trial of new drug, biochemical tests are very important. Biochemical investigation hold the key for the diagnosis and prognosis of diabetes mellitus, jaundice, myocardial infarction, gout, pancreatitis, rickets, cancer, acid-base imbalance. Successful medical practice is unimaginable without the service of clinical biochemistry laboratory. Means without the biochemical test or without the presence of the biochemistry laboratory, we can't diagnose and prognosis this disease. This all diseases like diabetes mellitus, jaundice, myocardial infarction, gout, this all are identified. This all are identified by using the biochemical or biochemical test biochemical tests that are present in the biochemistry laboratory. So without the biochemical test, we can't investigate the disease, right? So biochemical tests are most important in the clinical laboratory. Clear? Now, what is the uses of biochemical test? What is the uses of biochemical test? The first use of biochemical test in the diagnosis. What? In the diagnosis. First use. Which type of diagnosis? The diagnosis of disease like diabetes, jaundice, gout, etc. So this type of disease are diagnosed by the biochemical test. Next, function and use of biochemical tests is screening the disease. How we screen the disease? Which type of disease is present? Which type of disease is present? These are identified by the biochemical test. Third is monitoring patient or assessing the prognosis or once a diagnosis has been made. When biochemical tests are performed and diagnose the disease, which type of disease is present in the person or person suffering of which type of disease are identified by the biochemical test. After that only patient are monitored. What type of drugs or medicine are provided to the patient after when diagnosis has been made. Next is research studies and in clinical trial of new drug. Already I told you in the previous slide, right? Already I told you in the previous slide that for the research also, biochemical tests are used for the investigation of the novel drug. Right? Clear? Now, what are the sample? What are the sample used for the biochemical investigation? Right? What are the different type of sample used in the biochemical test? The first sample is the blood, right? Second is the urine. Third is the body fluid, and fourth is the cerebrospinal fluid. You know, every person once a life go go in a biochemical laboratory for the examination of blood, right? So during that technician came and collect a sample, that is blood. Why they collect blood? Why they collect blood? Because in our blood everything are present, like antigen, antibody, carbohydrate, lipid, all things are present in our body, sorry, in our blood, because blood is consist of serum, plasma, whole blood cells, and these are separated by a analytical techniques, that is centrifugation. Which technique? By using the technique centrifugation. So serum is a clear liquid that can be separated from the clotted blood. It does not have fibrinogen or the other clotting factor. Plasma is the liquid portion of the blood obtained by centrifugation of blood treatment with anticoagulant. And whole blood cell are whole blood cell. So for the identification of the antigen, antibody, carbohydrate, lipids, blood sample are collected. Next is urine. What? Next is urine. For the know the to know the functioning of the kidney, kidney function is proper or not, the urine sample is taken. What? Urine sample is taken. Third is body fluid, and fourth is cerebrospinal fluid. Cerebrospinal fluid are taken for the identification or know the proper functioning of the brain. Cerebrospinal fluid is used as a sample. So these four are the samples that are collected by the technician for the identification of the biochemical test, like blood, urine, body fluid, and cerebrospinal fluid. So first test that we study in this video is the GTT. The expand form of GTT is glucose tolerance test. What? The expand form of GTT is the glucose tolerance test. From name it is clear. What? From name it is clear that this test is for the identify the level of the glucose. Identify the glucose level. Right? So GTT, short for the glucose tolerance test, is a test designed to assess the body response to glucose. What? GTT is designed to assess the response of body on the glucose. How our body will respond? to glucose. When we eat the food and the glucose that are present in that food, how our body responds to that glucose. Insulin are produced in response to that glucose. If the insulin are produced, then how much insulin are produced? So this all are identified by the glucose tolerance test. Next, in GTT, the patient given a glucose solution. What? Glucose solution and blood samples are drawn afterward at interval to measure how well the body cells are able to absorb glucose. How glucose are absorbed in our body with the help of enzyme, that is insulin, right? That are secreted by the pancreas. That are secreted by the pancreas. So insulin can absorb the glucose in the body that is secreted by the pancreas. Generally, GTT tests are performed in a pregnant woman. There are several variations to the glucose tolerance test used in different conditions, but the most common one of them is the oral glucose tolerance test, that is OGTT. The OGTT is mainly used in the diagnosis of gestational diabetes. For the diagnosis of oral glucose tolerance test are used for the diagnosis of gastrointestinal diabetes. For oral glucose tolerance test, the patient is required to fast for 8 hours. What? 8 hours. And then a fasting plasma glucose is tested. After that, oral glucose solution is given. This is performed in morning mainly. Mainly this OGTT is performed in morning condition. When in night, when a person eat and sleep, after 8 hours, when they wake up in the morning, the OGTT sample will be drawn by the technician and test for the glucose tolerance test, how our body responds to the glucose. After that, blood sample can be drawn up to 4 times at different intervals to measure the blood glucose. Now, who is given glucose tolerance test? For whom it is used? Already I told that GTT is used for the pregnant woman. The GTT is usually given to pregnant women during 24th and 28th week of pregnancy. This test is also given to pregnant who has diabetes symptoms or have the risk of developing diabetes prior to the pregnancy. Actually, nowadays GTT is used by all the pregnant women. They are diabetes or not. These tests are performed by the all pregnant women in 24th and 28th week of pregnancy. But for the person who have already diabetes, for that person, this GTT is compulsory. What? Mandatory. This GTT for the diabetes person. Besides that, the GTT is also given to other patients who are experiencing symptoms of varied diseases that can cause high glucose level in the blood stream or restrict the proper absorption of glucose by the body. If our body not absorb the glucose, right, or cause a high glucose level in the blood, that patient also GTT are performed to know the functioning of the body in response to glucose. People with a high level of sugar have the following side effect in response to drinking the glucose. When they drink the glucose, because in GTT before the testing, few hours before the testing, glucose is taken by them. So when they take the glucose or drink the glucose, if they have a high level of glucose, then the body will respond like they having a symptoms like nausea. What? They having the symptom of nausea, stomach discomfort, diarrhea, constipation. Body will respond nausea, stomach discomfort, diarrhea or constipation. Right? Now what is the normal value of the GTT? The GTT normal value is lower than 140 mg per 140 mg per dl, and if the blood glucose level between 140 to 199, this is the normal value. The GTT normal value is the normal value is less than 140, less than 140, or if in between 140 to 199, means they are a strong indicator. What? They have a strong indicator of pre-diabetes. This is the value at the pre-diabetes. OGTT and oral glucose tolerance test normal range for fasting result is 100 to 125 mg per dl, and for pre-diabetes it is 126 or greater for diabetes. If more than 126 means the person having a diabetes, and greater than 92 means they are gestational diabetes. Greater than 92 means they have gestational diabetes. The OGTT normal range for after 2 hour test result in between 140 to 199 for pre-diabetic, already I told, and 200 or greater than 200 for diabetes, and greater than 153 for gestational diabetes. Remember this all the value, because this value are only for learning. So remind this value. This is very important. Next is lipid profile test. A lipid panel or lipid profile test assesses the risk for cardiovascular disease in a person. What type of risks are caused by the lipid profile? Cardiovascular disease. It is a combination of various blood tests which is performed to measure the level of 4 types of lipids in our blood. What? Measure the level of 4 types of lipids in our blood, such as total cholesterol, low density lipoprotein cholesterol, high density lipoprotein cholesterol, and triglyceride. SDLC, right? SDL, LDL cholesterol, triglyceride, and total cholesterol. These are included in the lipid profile. Remember this. What type of 4 different type of tests are performed in lipid profile test? Total cholesterol, LDL cholesterol, SDL cholesterol, and triglyceride. Clear? Now next is a lipid profile test is required to identify the symptom associated with dyslipidemia or abnormal level of lipid. Some of the symptoms of dyslipidemia are upper back pain. What? Upper back pain, hypertension, general malaise, feeling of discomfort, chest pain, shortness of breath. Shortness of breath is the most common factor. Heartburn, arm pain. Arm pain is also a most common factor for the abnormal functioning of the body in response to lipid. In response to lipid. What is the procedure for the lipid profile test? The doctor may ask you to fast. The doctor may ask you to fast before undergoing a lipid profile test. The sample are taken after 12 to 14 hours of overnight fasting so that the result will be more accurate. Means we have to fasting overnight for the lipid profile test so that we get a more accurate result, test or result. You can drink clear fluids such as water, but you should avoid drinking beverages such as coffee and tea. Before the lipid profile test, you need to take certain precaution. You need to avoid drinking alcohol, avoid high fat food, avoid strenuous exercise. This all three steps are avoided. You actually you only drink water from before 12 to 14 hours. Before 12 to 14 hours. The sample for the lipid profile test taken by a technician is the blood. What? Blood. Blood is taken as a sample for the lipid profile test. Now next is liver function test. Liver function test is performed for know the proper functioning. To know the proper functioning of the liver. So liver function test is performed. LFT means a number of tests to assess the various function performed by liver. The important among liver function tests are total protein, albumin, globulin, bilirubin estimation, SGPT and SGOT. These are the most important factor that are tested in liver function test. Estimation of bilirubin. There are three different methods by which bilirubin are estimated. First is Malloy and Evelyn method. Second is DMSO method. And third is Groff method. These three are the method by which liver functioning tests are estimated. When we go for a liver function test and when a doctor perform or recommend you to go for liver function test, if you have a jaundice, dark urine, you have a nausea, vomiting, or you are tired or weak, then doctor recommend you to go for the liver function test to determine when certain drug treatment should be started. To monitor the progression of a liver disease. Monitor the progression of a liver disease. How the liver disease will be considered. These all are decided by a liver function test. To assess your response to a liver treatment. How our liver will respond when we go through the treatment. For that also doctor recommend for the liver function test. What is the routine liver function test? Liver function tests are blood test. Sample, what sample doctor takes? Doctor take a sample, that is a blood. So liver function tests are blood tests used to diagnose and monitor disease or damage of the liver. What? They monitor the damage of the liver. And the sample from the blood that was isolated and tested by the technician are the serum albumin. What? Serum albumin. Second, the blood liver enzyme. The enzyme is alanine aminotransferase, aspartate aminotransferase, gamma glutamyl transferase, alkaline phosphatase. Third is blood bilirubin. And fourth is blood coagulation factor, that is prothrombin, right? So these four are the that are a molecule or a sample that are present in blood that are identified or tested in a liver functioning test. Next is renal function test. What are the renal function tests? The major function of the kidney are to excrete metabolic waste product as well as to maintain water, pH, electrolyte. What? And to maintain water, pH, electrolyte balance, production of calcitriol and erythropoietin in the liver or our body. So major function of the kidney is to excrete basically metabolic waste product and maintain the water pH electrolyte balance, right? A decrease in kidney function is due to a reduction in the performance of the nephron. Already you know that this is the glomerulus. Now these are a shape. How the kidney, how the electrolyte balance will be takes place, right? And how the water and this all are the maintain a balance, right? So these are the nephron. What? Nephron. They maintain the proper balancing the function of the kidney. So if these nephron are not working proper, means there is something went wrong to the kidney. The functional unit of the kidney is the nephron. What? Nephron is the functional unit of the kidney, which is composed of the Woman capsule. This is a Woman capsule with the glomerular tuft of capillaries, right? The proximal convoluted, which is composed of the Woman capsule. This is a Woman capsule with the glomerular tuft of capillaries, right? The proximal convoluted, this is a PCT, this is the DCT, this is the DCT, and collecting tubules, right? So this is the parts of kidney by which the water pH electrolyte balance will be takes place. Clear? Next is the function of kidney at a glance. Maintain the, already you know what is the function of the kidney. Kidney maintain the electrolyte balance, excretion of urea and other waste products such as creatinine. Creatinine, remember this. When kidney not function properly, then this creatinine go in the blood. And if the level of the blood, creatinine will increase in the blood, means they act as a toxic or poison. So for the removal of this creatinine from the blood, artificially dialysis process will be performed. So such as creatinine, these are a waste product, uric acid and metabolites of xenobiotics. These are a waste product. Next, maintaining water balance. Fourth, excretion of sodium, excretion of potassium, effect on heart, excretion of hydrogen ion that maintain the pH. So these are the function of the kidney. Now what are the symptoms when our kidney not perform properly? When our kidney not perform properly, the symptom is fever, stomach pain, vomiting, dizziness, blood in urine, and back pain. These are the function of the symptom of the kidney when they are not performed well. Next is urinalysis. How they examine the urinalysis? There are three methods. First is physical examination, microscopic test, and biochemical examination. In physical examination, we examine the volume, color, order, reaction, and specific activity. Volume means how much water you drink and how much water will excreted by the body. Color of the urine, order, these are the physical examination. In microscopic test, we examine the urine in the microscopic to see the cells, crystals, casts, microorganisms, parasites, and contamination. And during biochemical examination, they identify the protein, sugar, ketone bodies, bile salts, bile pigments, and bloods. So these are the examining how the urine are examined in physical examination, biochemical examination, and microscopic examination. Next is renal function test classification. Urine analysis, already I told you, physical examination, chemical examination, and microscopic. Next is assessment of glomerular function. In this, renal clearance test will take present, blood analysis of urea and creatinine, proteinuria, and hematuria. Third is test to measure the renal plasma flow. Fourth is test for assessment of tubular function. These all are parts of the kidney, nephron, that is PCT, DCT, collecting tubules, right? So these all are tests for different parts of the kidney, nephron. Urine concentration test, urine dilution test. Next, fifth is renal biopsy to confirm the diagnosis and renal disease. Which type of disease are present in a kidney. Test for tubular function, a specific gravity of urine. Normally, specific gravity of urine is 1.015 to 1.025. This is an indication of osmolarity. If a specific gravity will decrease, excessive water intake excessive water, and diabetes insipidus are present. If your specific gravity will increase, means diabetes mellitus is present. Nephrosis, renal failure will be occur sometime. So these are the kidney function tests. The tests that are part of the kidney function test panel are urine examination, serum urea, serum creatinine, blood urea nitrogen, calcium, phosphorus, protein, albumin, creatinine clearance, urea clearance, inulin clearance, and dilution and concentration test, serum electrolyte level. So these we identify in a kidney function test. So these all are biochemical tests that are covered in this PPT, in this video. So today we discuss about the IMVIC test that comes under the Bacteriology 403. What happened in this test? Mainly this test is for Enterobacteriaceae group. So this test is a combination of 4 types of test, that like indol test, methylrate test, citrate test. So this is the combination. This test is a combination of multiple tests. So first, what is the introduction? The IMVIC tests are a group of individual tests used in microbiology lab testing to identify an organism in the coliform group. Mainly this test confirm the coliform group of bacteria, right? So coliform, what is a coliform? Coliform is a gram-negative. What? Gram-negative aerobic bacteria or facultative anaerobic rod which produces gas from toes within 48 hours. What? In 48 hours. The presence of some coliform indicates fecal contamination. Mainly this coliform group of bacteria are present in fecal contamination. So if these bacteria are present in the sample, it means there is a fecal contamination are present. Right? So IMVIC test are a group of individual tests used in microbiology lab which confirm the organism like coliform group. Clear? Which confirm the organism like coliform. So the triple sugar iron test. It is a triple sugar iron test. Urease and malonate test are also included in this test. Already I told you that IMVIC test is a combination of several types of tests, right? IMVIC test is a combination of several types of tests. These tests are useful in distinguishing members of Enterobacteriaceae. Mainly these tests are used for Enterobacteriaceae group of the bacteria that confirm the gram-negative bacteria, right? That gram-negative bacteria like E. coli, or they confirm the fecal contamination. So the principle of IMVIC test, each of the letter in IMVIC stand for one of this test. Means I stand for indol. In IMVIC, I stand for indol. M is for methyl, and V is for Voges Porescore, and C is for citrate test. And this lower I, right? This lower I indicate added for the case of pronunciation for only, right? So this is the IMVIC test. I stand for indol, M stand for methyl red, V stand for Voges Porescore test, and C stand for citrate test. So it's a combination of several types of test. IMVIC test is the combination of several types of test. So IMVIC is an acronym that stands for 4 different type of tests, that is indol test, methyl red test, Voges Porescore test, citrate utilization test, right? It is a combination of 4 types of tests. So we discuss this all the 4 types of tests individually in detail. So first we discuss the indol test. A series of 4 tests consisting of indole. Indole, what happened in indole? In indole, they break down the amino tryptophan. In indole test, tryptophan are breaking down. And methyl red, there is oxidation of glucose. Glucose oxidation takes place in the methyl red test. Clear? And in Voges test, production of neutral and product. Production of neutral and products. And citrate is the fermentation, citrate fermentation. It is used to differentiate the Enterobacteriaceae, and we look at each test individually. So to obtain the result of these 4 tests, 3 test tubes are inoculated, that is tryptophan broth, because in indole test what happened? There is breakdown of the tryptophan will be takes place. And in methyl red test, there is oxidation of methyl group. Clear? So in this what happened? There are 4 actually in this IMVIC test 4 different tests are present. So in this 4 different test tube we present 1, 2, 3, 4. And in this test, individual test will be takes place, and according to the color we decide IMVIC tests are positive, indole tests are positive or negative, methyl tests are positive or negative, or citrate tests are positive or negative. So according to their color or presence of a circular ring, we decide the positive or negative, right? So IMVIC tests are employed in the identification and differentiation of membrane of Enterobacteriaceae, family Enterobacteriaceae. So first indole test. What is the principle of first indole test? Certain microorganisms can metabolize tryptophan by tryptophanase. This is the enzyme. Tryptophanase is an enzyme. The enzymatic degradation leads to the formation of pyruvic acid, indole, and ammonia. When the enzymatic degradation of the tryptophan leads to the pyruvic acid, indole, and ammonia. The presence of indole is detected by addition of Kovacs reagent. What is detected by addition of Kovac reagent? Clear? The presence of indole is detected by addition of Kovac reagent and tryptophan amino acids. These tryptophan amino acids are break down into indole plus pyruvic acid plus ammonia. And the enzyme which degrades this tryptophan is tryptophanase. What? This is the enzyme, that is tryptophanase. This enzyme break the tryptophan amino acids. And this is the Kovac reagent. What? This is the Kovac reagent which produce red color in upper organic layer. And this red color decided the positive or negative. It is performed on sulfide indole motility, that is SIM. What is sulfide indole motility medium, or in tryptophan broth, or in motility urease indole MIU medium? Result is read after adding the Kovac reagent. When we add this Kovac reagent, after that we read the result. The positive result indicated the red layer at the top of the tube after the addition of Kovac reagent, and negative result is indicated by the lack of color change. What? A negative result is indicated by the lack of color change at the top of the tube after the addition of Kovac reagent. So this is the procedure. How this test will be takes place. See, this is the indole test result. This is the red color will be present as the top. This indicates the positive result, and this having no red color. This is the negative result. So we comes under the procedure. What the procedure will be takes place. Bacterium to be tested is inoculated in a peptone water which contain amino acids like tryptophan and incubated overnight at 37 degrees Celsius, and prepare 1% tryptophan brosate 1. Okay. Incubate one set of test tube with test organism and maintain one set as negative control, and without means negative control means without inoculation. Negative test means without inoculation, and inoculate one set of test tube with E. coli. Use a positive control. Use as a positive control. And following incubation, few drops of Kovac reagents are added. Few drops of Kovac reagent are added. Shake gently. Kovac reagent consists of para-dimethyl amino-benzaldehyde 10 gram and isoamyl alcohol 150 gram and concentrated HCl 50 ml. In somewhere, some question will ask, what are the Kovac reagent and what the material, what the solution and what are the reagent by which this Kovac reagent are made, right? Then this Kovac reagent are consist of para-dimethyl amino-benzyl aldehyde and isoamyl alcohol and concentrated HCl. Remember this. Kovac reagents are made up of para-dimethyl amino-benzyl aldehyde, isoamyl alcohol and concentrated HCl. Clear? Allow the tube to stand for 2 minutes. So that reagents come through the top and culture with the control tube. See this. The reagent will comes at the top. This is known as the indole positive, and when reagent comes at the top and produce red color, and if the reagent are not come at the top, then this is the indole negative. This produces indole negative test. This is a methyl red test. Next is a methyl red test. What happened in methyl red test? We add the reagent, that is the methyl red reagent, and when we added the methyl red reagent, there is a development of red color after the addition of methyl red, and a negative methyl red test is indicated by no color change after the addition of methyl red reagent. See here, this is the addition. When we add the methyl red, they produces a color. We added in both, right? In both the test tube, in this also and in this also. When we add the methyl red, then if this sample having a positive methyl red test, then they produce a red color, and if they are negative, then there is no change of the color. So the procedure is the bacterium to be tested is inoculated in the glucose phosphate broth, which contain glucose and phosphate buffer, and incubate at 37 degree for 48 hours. Over the 48 hours, the mixed acid producing organism must produce sufficient acid to overcome the phosphate buffer and remain acid. The pH of the medium is tested. The pH of the medium is tested by the addition of 5 drops of methyl red reagent, and there is development of red color is taken as positive. This red color is taken as positive. MR negative organisms produce yellow color. The methyl red negative organism means the organism which not contain glucose phosphate broth, that organism produce yellow color instead of red color, right? And the organism which contain the glucose phosphate, that organism produce red color. Clear? Next is VP test. VP test is the negative test is indicated by a lack of color. Negative test is indicated by a lack of color change after the addition of Brits A and Brits B reagent. Remember this. What? Brits A and Brits B reagent. A positive Vogel's proscusis test is indicated by the development of red-brown color after the addition of Brits A and Brits B reagent. A positive Vogel's proscusis means VP test is indicated by the development of red-brown color. See, in indole test also and in methyl red test also, in both tests, the test tube after the addition of coact and methyl red reagent, there is production of red color, right? There is production of red color at the top of the test tube. Whereas, but in VP test, there is the production of red brown color at the top after the addition of Brits A and Brits B reagent. Clear? When we add coac reagent in indole test, in previous slide we see that we add the tryptophan solution. When tryptophan is present, enzyme which break down the tryptophan into the pyruvic acids and ammonia, then that pyruvic acid, when we add the coac reagent, that will be converted into a red color, right? That produces the red color, right? So that in that red color will be produced. Similar when we in methyl red test, when glucose phosphate is present in a culture and we add the methyl reagent in that, then that will also produces the red color. Clear? That will also produces the red color. So methyl red test and the indole test, both are produces red color, and if sample is negative, then there is yellow color will be remaining the yellow color, right? But in VP test, when we add Brits A and Brits B reagent, there is production of red brown color. What? Red brown color. You have to remember the reagent, which reagent we added in which type of test. Procedure. Bacterium to be tested is inoculated into glucose phosphate broth and incubated for at least 48 hours. 0.6 ml of alpha naphthol is added to the test broth and shaken. 0.2 ml of 40% potassium hydroxide is added to the broth and shaken. The tube is allowed to stand for 15 minutes. The tube is allowed to stand for 15 minutes, and appearance of red color is taken as a positive test. Means red color, appearance of red brown color is taken as a positive test. The negative tube must be held for 1 hour, since maximum color development occurs within 1 hour. Addition of the reagent, that is the Brits A and Brits B reagent. This is the test tube. When we added the reagent of the Brits A and Brits B reagent, we added, they produces a red brown color. See, this is the red brown color. This is the positive VP test, and this is the negative VP test. In this, there is no addition or production for red brown color. Now, citrate utilization test. Last one is IMVIC test is a combination of 4 types of test, right? In 4 type of test, we already discussed about the 3 types of test. The last one is citrate utilization test. So, in this test, the test is performed on Simon citrate agar, in which the test is performed on Simon citrate agar. Negative citrate utilization test is indicated by the lack of growth and color change in the tube. Negative citrate utilization test is indicated by the lack of growth and color change in the tube. A positive citrate result as indicated by growth, the blue color change. Positive citrate indicated by the growth and blue color, right? And negative citrate test is indicated by the lack of growth and color change in the, right? Remember the color in citrate test. Positive indicated by blue color and negative indicated by there is no change in the lack of growth and color change in the tube. Okay? So So what is the procedure? Bacterial colonies are picked up from a straight wire and inoculated in a slope of Simon citrate agar. Suppose this is a plate of agar, right? This is a plate of agar. In this, from a loop, we take a bacteria colonies from the loop and we produces a straight wire like this, right? If the organism has ability to utilize citrate, the medium changes its color from green to blue. Then if this agar contain a citrate, right? This agar contain a citrate. If this agar having a citrate and the bacteria or organism we placed in this agar, bacteria or organism placed in this agar, then this if bacteria this bacteria utilize this citrate which are present in the agar, then the color of the plate or test tube will be changes from green to blue if this bacteria utilize the citrate that are present in the agar. Observation: if color of the medium changes to blue, it is citrate positive E. coli, right? It is the citrate positive E. coli. See, this is the plate. This is a test tube which contain the citrate agar. When we placed or inoculate this test tube with the bacteria, and that bacteria utilize this citrate. If that bacteria utilize this citrate, then the color of the citrate will be changes from blue, right? So in this what happened, this is the green color citrate, there is no change in color, means this is a negative result, and this is the positive result because it is in this there is utilization of the citrate. So in vivo test result of genera of Enterobacteriaceae. For E. coli, in this we discuss about the for the different type of microorganism which microorganism produces which test positive and which test negative. So first we discuss for the E. coli. For E. coli, in vivo test, indole is positive, means they produce red color. Methyl red is positive, means they produce red color. VP test is negative, means they not produce red brown color, right? Citrate test is negative, means they not utilize the citrate and not there is change of the color from there is not change of the color like from green to blue, right? So this test is negative. Imbic test for E. coli is plus negative, means all test is positive, methyl red test is positive, VP test is negative, citrate test is negative, right? Imbic test for Klebsiella. In this, indole test is negative, means there is no production of red color, means the tryptophanase enzyme are not present which break the tryptophan into pyruvic acid, because Kovac reagent only change the color when there is presence of pyruvic acid. If pyruvic acid they react with the pyruvic acid, Kovac reagent, right? So tryptophanase enzyme are not present. Its tryptophanase enzyme are not present. Methyl red test is negative. Methyl means there is no oxidation of glucose will be takes place. VP test is positive. VP test positive means there is production of red brown color. Citrate test is positive, means in this there is presence of bacteria which utilize the citrate, right? That's why it is the positive. See, this is the test tube of the INVIC test. See, this is the indole test having positive and this is negative. Methyl test positive and negative. VP test See, this is positive and this is negative, and this is citrate agar test. Citrate agar test, what happened? This is the agar in test tube. They are having the agar, and this agar, what happened? What happened by this agar? This agar changes the color of green citrate into a blue color. Means this is the positive, this is the negative. So combination of this all test is known as IMVIC test. What? IMVIC test. Combination of this all test is the known as IMVIC test. This test is mainly used for the family of Enterobacteriaceae and coliform bacteria, which confirm the fecal contamination. This is the IMVIC reaction. See, for E. coli, IMVIC test is positive and methyl red test is positive, but VP test is negative and citrate test is negative. For Edwardsella tarda, IMVIC test is positive, methyl test is positive, same as E. coli. And for Proteus vulgaris, same as E. coli. Klebsiella pneumonia, for this IMVIC test, indole test is negative, methyl test is negative, VP test is positive in this, and citrate test is positive, right? Klebsiella oxytoca, in this Klebsiella oxytoca, IMVIC test is positive, methyl test is negative, VP test is positive, and citrate test is positive. Enterobacter species, in this also indole test is negative, and this methyl test is also negative. VP test is positive, citrate test is positive. Serratia marcinus, Serratia, in this is also like this Klebsiella pneumonia. The indole test and methyl test are negative, whereas VP and citrate test are positive. Citrobacter, from name it is clear, Citrobacter means they having a citrate, right? So this citrate test is positive, and citrate test in this is a different from all of this, means there is in this what happens, citrate test is positive, methyl test is positive, indole and VP test is negative. And for Citrobacter, in Citrobacter, in indole test, methyl test and citrate test, these three are positive and VP test is negative. Clear? So what is the application? Application of the IMVIC test. The IMVIC tests are a group of individual tests. What? IMVIC tests are a group of individual tests that is used in microbiology lab testing and testing to identify an organism in a coliform. Mainly it is used to identify a coliform bacteria or a family of Enterobacteriaceae. What? Or a family of Enterobacteriaceae. Right? Coliform is, what is the coliform? In starting slide already I discussed what is the coliform. Coliform is a gram-negative aerobic bacteria. What? Gram-negative aerobic or facultative anaerobic rod-shaped bacteria, which produces gas from lactose within 48 hours. The presence of some coliform indicate fecal contamination. What? Presence of some coliform indicate fecal contamination. So in this all type of, see this, in this what happened, which type of reagent are used, and what type of reaction will be taken. In last we summarize this all, right? What type of reaction will be takes place in this all four different type of test, and which type of reagent will be used. So in indole test, what first IMVIC test is a combination of four types of tests, that is first indole test, methyl red test, VP test and citrate test, right? So first indole test. In indole test what happened, there is the breakdown of the tryptophan, tryptophan, that is the amino acids, and converted into a pyruvic acid with the help of enzyme tryptophanase. With the help of enzyme tryptophanase. And a Kovac reagent are used in this reagent. In this test, which type of reagent? Kovac reagent. Remember this. In doll test, which reagent are used? Kovac reagent, which changes the color reactive pyruvic acid and changes the color into red, right? Whereas in methyl red test, what happened? There is oxidation of glucose phosphate, and when glucose phosphate oxidation of glucose phosphate will be takes this, we added the methyl red reagent, and there is a production of red color, and this red color if produced means there is a called as a positive test. In VP test, what happened? Reagent group A and B are used, right? A and B are used, and in this what happened? If that reagent produces red brown color of the colonies, means these are a positive test. Clear? In citrate test, we take a citrate agar solution. What? We take citrate agar solution, and we inoculate the bacteria in this test tube, in this agar test tube, and if that bacteria utilize the citrate, then there is a change in the color, that is blue, and if that bacteria not utilize the citrate, then they remain as a green. So blue color is a positive result. What? They indicate the positive result. So these are the combination of these 4 types of test, right? Indic test is a combination of 4 types of test. Remember this, and this is mainly used for the family of Enterobacteriaceae. Which family? This is mainly used for the family Enterobacteriaceae. So this all about this IMVIC test. So today we stop here. Hope this video are clear to all. Okay, thank you.